Thank you for your interest in my poster entitled Development and Validation of LC-MS/MS Assay Methods to Determine ubrogepant and atogepant in Human Breast Milk, presented at the AAPS 2024 PharmSci 360 in Salt Lake City, Utah. My name is Mathieu Lahaie, and I am a senior research scientist in method development at Altasciences. I will provide you a brief overview of the work we have conducted. Ubrogepant is used for the acute treatment of migraine, while atogepant is used for the preventive treatment of episodic migraine in adults. Studies in pregnant women indicate that migraine often improve or disappear during pregnancy, but tend to reappear after birth, making migraines a concern in lactating women. The pharmacokinetic of ubrogepant and atogepant in lactating women and their excretion into breast milk have not been evaluated yet. Consequently, the lack of evidence regarding infant exposure to ubrogepant or atogepant through breastfeeding may prevent patient usage of these drugs. A novel LC-MS/MS bioanalytical method was developed to determine the excretion of ubrogepant and atogepant in breast milk. The method consists of a protein precipitation extraction, and the samples were injected on an Agilent 1100 HPLC system coupled to a SciEx API 5000 mass spectrometer using positive electrospray. Chromatographic separation was performed in reverse phase mode using a Waters-X-Bridge C18 column with an acidic mobile phase. The chromatography was also optimized to flush the column with high organic mobile phase and flow rate at each injection to avoid late elution interference and MS signal suppression from the phospholipids. This allows method reproducibility and robustness. The extraction recovery was quantitative for both ubrogepant and atogepant. Calibration curves were linear from 1 to 1000 nanogram per ml, where the precision was less than 10% and the accuracy was within 91 to 108%. Specificity, matrix effect, and matrix factor for ubrogepant and atogepant all meet acceptance criteria according to the ICH guideline M10, based upon control from 6 individual breast milk donors. Ubrogepant and atogepant were stable at 4 degrees Celsius in breast milk for at least 25 hours. The stability storage at minus 80 degrees was 240 days for ubrogepant and 83 days for atogepant. Both compounds were also stable with 4 freeze-thaw cycle, and the post-extraction stability was established for 168 hours at 4 degrees Celsius. In conclusion, the method developed for the quantitation of ubrogepant and atogepant in human breast milk was successfully validated and demonstrated to be specific, precise, and accurate, making it suitable for clinical sample analysis.